it's not as rich as you think it would be. But think about the fatty acids, right? Those little cells, they could be a little fluid in motion, right? And so when looking at this, the proteins move freely for various functions. The phospholipids, they rotate and they move laterally, right? And self-healing is good because if there's a tear in the plasma membrane, the plasma membrane can heal itself, can like amend that tear and bring those membranes a little bit tighter, bring those phospholipids a little bit closer to self-heal. Alright, so that's great. But the thing that's really important for the plasma membrane is that it has selective permeability. I would highlight that term because that means it allows the passage of some molecules but not for other molecules to pass through there. Right, so some things can pass through there, some things not so much. And it makes sense to where if you look at it, you can say, okay, well, yeah, water can pass through the cells because water has a little charge on it, right? It can interact with the phospholipid head of that particular phospholipid bilayer. And so something bigger things need some type of channel to like come inside. It can't just pass through there. Now, when talking about the plasma membrane, it also has enzymes for ATP production, right? So some of those proteins that could be there could be just for ATP production. We know ATP production is important for making energy for the cell. Now, some membranes have photosynthetic pigments. What cells would have photosynthetic pigments in the membrane? I heard it. The chloroplasts are going to be your organelles. So within your plasma membrane, which is that external component, right? What type of cells would have the photosynthetic pigments in this plasma membrane? You're on the right track.
It's for plant, right?
Plants, there we go. So not chlorophyll, it's plants. Right? So plants have photosynthetic pigments in their cell wall, or their plasma membrane. And so some membranes have these photosynthetic pigments on their photos called chromatophores. Alright, chromatophores are going to be those for references to photosynthetic pigments. Alright, so let's look at this little video real quick. Well, first look at the chromatophore. So that's what the chromatophores are going to look like within the structure. But I want to go back and let you guys listen to this video on how things pass in and out of the cell.
Hydrophobic pores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
All right, any questions? Was it a lot? Was it easy? Simple to follow? Alright, you have questions? Let me know. So, when talking about the plasma membrane, I kept mentioning that the antimicrobial agents, we talked about antibiotics. Antibiotics can destroy the plasma membrane. Well, cell wall. So antibiotics can destroy the cell wall, right? But there are some antibiotics that actually will target the plasma membrane itself. So let's look at disinfectants first, right? So disinfectants we know are used to clean what? Surfaces. Can you wash your hands with disinfectants? No, don't do that. You can, but it's not a good idea. You're gonna kill all the good bacteria, so please don't do that, alright? So disinfectants, they can damage the plasma membrane. So alcohols are really good for that, right? So our quaternary ammonium compounds, aka detergent that you use to wash your clothes with, right? So that's why you wanna make sure that you wash your clothes because of removing those different potential pathogens or even just microbes in general. Not always pathogenic, right? So then you have antibiotics. You ever heard of Neosporin or triple antibiotic ointment? Triple antibiotic ointment, yeah. So triple antibiotic ointment is going to include the antibiotic or polymyxin. Alright? So that's why we're talking about triple antibiotic ointment. It's going to include neomycin, polymyxin, and bacitracin. All three of those things target different parts of the bacterial cells. So polymyxin targets the plasma membrane. Alright, I'll tell you guys as we talk about it throughout the semester, how that comes out. That's why it's kinda good sometimes to put if you cut yourself, you hurt yourself some type of way, your skin is open to the surrounding environment, clean it, put some ointment on it, so that way it can prevent the actual infiltration of bacteria, right? You don't want the cut to become infected with a bacterial infection, right? So that will allow for that from happening, but also don't apply neonates or triple antibiotic ointment as a preventative either, because it's still an antibiotic, alright? So that's one thing I try to tell people, like, you don't wanna just I know people that were putting, triple antibiotic ointment in their nose during COVID, I'm like, don't do that, you still not get that serious, right? Don't just do it as a preventative, only if there's an actual, lesion or wound or something to protect, alright? And then lastly, damage to the plasma membrane, of course, will cause the cell contents to leak out, right? That's the barrier that protects it or, basically separates it from the outside of the cell and the internal components of the cell. So everything will pour out if you damage the plasma membrane. Alright, this is fun. You've learned about this before, right? Passive processes, passive diffusion, passive transport, active transport. So passive is basically where it naturally happens. It naturally happens. There's no energy needed for it, it just don't happen. So substances will move from an area of high concentration to an area of low concentration naturally. Whereas active transport is gonna go against that, it's the opposite. Alright, and because this is an unnatural process, it doesn't naturally happen, there needs to be an energy input for it to happen. So for active, it's gonna be substances going from low to high. Alright, so passive is from high to low, it makes you, so passive, if it helps, some people need to like see it written out. So passive, high to low. And then for active, you're gonna have from low to high. And from low to high, you're gonna need an input of energy. And so this energy is gonna come in the form, ooh, that's a bad little star. Close, ATP. Looks like Patrick Star. Okay, ATP. Alright, so you need some energy for that one, ATP. Alright, so let's look at passive, the easy one. So this is the one that naturally happens. So simple diffusion is when there's gonna be a solute. We talked about solutes, right? So solutes are typically added to combined with the solvent to be dissolved and to make it a solution, right? And so basically the solute moves from an area of high concentration to an area of low concentration naturally. And that happens until equilibrium is established. That's the goal. We wanna have equilibrium, meaning the same concentration on both sides or both mediums, right? Doesn't mean the same number, I'm saying the same concentration, right? So oxygen and carbon dioxide, you guys saw those. So oxygen and carbon dioxide molecules just passing on through the plasma membrane. They just go through, their gases. They can pass through the membrane via simple diffusion, they're gases. They can just go. And that's great because you need carbon dioxide to stay in the process. And you need to release carbon dioxide as well, right? So works out perfectly. When looking at this figure 4-16, it shows us what would happen if we were to drop a dye of some sort into a beaker of water. Those particles are going to disperse into that water, right? And they ultimately dissolve and make that solution it will change colors. One example I like to use is perfume, right? You guys wear perfume, cologne, stuff like that? You guys wear that? I wear perfume. So with my perfume, if I decided to take my perfume bottle and bring one of my big bottles and sit it right here and open it, I'm gonna lose that whole perfume bottle because what's gonna happen is that the particles are gonna diffuse out of that perfume bottle to try to fill the whole room until equilibrium is established, right? This is a really big room, so I'm probably gonna lose that whole perfume bottle, right? In my house, though, it'll be a little bit different. So if I go to my house and I like go in my closet, well, one of the closets and like open that perfume, it's gonna diffuse out, but I may not lose my whole bottle, right? The particles may not diffuse all the way out, only because of the fact that the equilibrium may be established a little quicker. Space. Alright? So when looking at the process for cells, right, the lipid bilayer, the molecules right now when you look at it, I see a lot more molecules outside the cell than inside the cell. They're gonna have to move into the cell. Right? They're gonna move into the cell because they wanna go from high to low until it's equal and equilibrium is established. So with passive transport, these different processes, I'm not gonna watch the video only because I can explain it to you guys. So diffusion is basically particles diffusing across the membrane. So then you have facilitated diffusion. So facilitated means that something's gonna facilitate the diffusion of those molecules or help out movement of those molecules. And these will be these integral proteins, right? So integral proteins will allow substances to either go in or out of the cell, right? And so when looking at non-specific facilitated diffusion, this just means that anything that could potentially pass through there is gonna pass through there. So if it only takes those charged particles, and charged particles are gonna pass through that bad boy. It doesn't have to be a specific charged particle. It doesn't mean it has to be sodium, or it has to be potassium, it can be any charged particle, right? But this bad boy though is specific. So specific facilitating diffusion means that it has to be a specific compound, right? So there are things called like sodium potassium pumps. So sodium potassium pumps will only allow for sodium and potassium to pass through there, right? Because they're designated as sodium potassium pumps. So those are some people that are taking A&P anatomy already, you know about sodium and potassium pumps already. Those of you that have to take it, you learn about it, alright? So with osmosis, it's the movement of water, right? From the area of high concentration to the area of low concentration. So in the video, you saw the water was moving through the membrane without any type of like hope, right? The water was just moving through the membrane, but there are some times where you need these things called aquaporins. Aquaporins are found heavily like on kidney cells, right? Which makes sense because kidney cells, water, all that good stuff, urine, getting those levels in a reasonable range. So with the water molecules, many of them can pass through without any help. But some of them they need some type of transmembrane protein that allows for it to be passed or allows for it to actually come out. Right? So it allows for a higher concentration in the cell. So when talking about facilitated, I explained that already. So basically intramembrane proteins will assist or facilitate the movement of different substances. And so this is gonna be still, remember, passive transport is always going to be no energy involved. This is a naturally occurring process. No energy is needed for it to happen. It's going to happen. And I've already mentioned how you have non-specific and specific proteins. And so I mentioned the specific ones already, the non-specific already. And then we have the different types of diffusion and more, talking about osmosis. Alright, so the only thing that's kind of new from what I've been saying so far is that with osmosis, that's the word, aquaporins, right? So aqua, water, porins means there's gonna be an opening, a hole, or some sort, some type of channel. So aquaporins are how water molecules will move in and out the cell, in addition to them already moving, right, naturally through the membrane. But this is for more, allow for more to come in. Alright, osmosis. Alright, what about osmotic pressure? That's actually quite important because it's the pressure needs to stop the movement of water across the membrane, right? So, you probably remember learning about this in previous biology courses, right? And so when learning about this in previous courses, they typically use dialysis tubing. Have you guys did that experiment with dialysis tubing where you fill it up with solution? You didn't take biology 1, y'all took chemistry 3A. Yeah. So, if you would have taken biology 1, in the lab, what we do is we're gonna have a setup with a beaker, and in the beaker, we're gonna have like different solutions in the beaker. And so in the solutions, they could be pure water, it could be concentration of like salt water or sugar water, right? And so when looking at that example, you're gonna have your little cellophane bag, which is a dialysis tubing bag. And with the dialysis tubing bag, we just talked about with the kidneys, right? So the kidneys, people who have the misfortune of having kidney failure, kidney disease, they have to go through dialysis, right? They have to go through that support to help with their lungs, not lungs, not lungs, to help with their kidney function, right? To allow them to pick up the nutrients, to gain all those different fluids that they need, or even lose excess fluids they don't need. So with the dialysis processing, they use the cellophane sac. And so the cellophane sac, that allows for the water molecules to move through, but keeps the solute. Whatever the solute is, whether it's sugar, whether it's salt, it's gonna stay inside of that. The only thing that's gonna move is the water. So when looking at this example, we're gonna have a solution. This solution is mainly a water solution. So this is only a solution of water, and then so back on the inside of it is gonna be a mixture of sugar water, right? So sucrose molecules and water. So one thing that I know is I'm looking at this, where is the water concentration the highest? In the beaker or in the bag? The water concentration. In the beaker. In the beaker, because I have more water in the beaker. There's no sugar at all in the beaker, right? So since I have more water in the beaker, there's a higher concentration of water in the beaker and a lower concentration of water in the bag. The water wants to move from high to low, it's going into the bag. It's going into the bag. So that's why in this particular example, once equilibrium has established, it's going to increase the amount of water in that bag. So the bag was like kind of shriveled up down here, right? And you can see the water level's about right there. And so in this case, you kind of see the water bag or the cellophane bag has like increased in its size, and the water level is also increased to account for the movement of the water. And you still have those sucrose present, right? The sugar molecules present. So there's an equal concentration of water. It might not be the same number of molecules per se, but it's an equal concentration of water. Right? There's an equal concentration of water. So I have some terms that go with that. And how I remembered it back in the day, I was like, okay, I'm gonna put the picture quick. Alright, so for the isotonic environment, iso means equal. So isotonic solutions means that there is no net movement of water because the same concentration of water outside the cell is the same on the inside the cell. Right, so it's an equal concentration of water in both environments. Right, inside the cell, outside the cell. Alright, hypotonic. So hypotonic is when the water is gonna move into the cell. And when the water moves into the cell, it's gonna swell up. So hypo, hippo sounds a little bit similar, right? So hypo, hippo, they're gonna swell, be a little bit bigger in size because they're gonna take up that water. Now the thing is, with cells that have cell walls, right? So plant cells, fungi, and protists, and then bacterial cells, osmotic lysis is not really an issue for them, right? Because they have that cell wall to protect that cell from like bursting, taking up too much water, or from shriveling up too badly, from losing too much water. But us, animal cells, you don't have cell walls. So that's an issue. Osmotic lysis could be a thing. So osmotic lysis can happen if the cell takes up too much water and the cell wall becomes too weak to contain it, or it's destroyed in some type of way, damaged in some type of way, the cell's gonna rupture, it's gonna lyse. And then hypertonic, so hypertonic, lower, this is gonna have a lower amount of water inside the cell because the water goes again from high to low. Right, so the water moves out the cell into the surrounding environments, and when that happens, the cell kind of shrivels up a little bit, and it's gonna have plasmolysis. Alright, so plasmolysis is basically when your plasma membrane is going to pull away from the cell wall, and that can cause that cell to shrivel up and eventually die if the conditions are not restored. Alright, so plasmolysis will allow the cell wall to still be intact, but the plasma membrane shrinks away from it, and the cell can shrivel up and die. Definitely. So what about active transport? So active transport, this is when you need an input of ATP, right? You need an input of ATP. So this is what I'm gonna do. I'm going to fast forward a little bit, only because I wanna hit some of the main things. So before I stop us for getting into the review session, I do wanna hit these things. So cytoplasm is our fluid inside the cell, right? Cytoplasm is the fluid on the inside of the cell. And it's mostly water, 80% water, plus it's proteins, carbs, lipids, and ions. You're like, oh, we learned about that, right? So then you have DNA. Now, when talking about prokaryotic cells, your DNA is in the nucleoid region. It's not an organelle, it's a region, right? A nucleoid region. You also have ribosomes. So ribosomes are dispersed throughout the cytoplasm, right, for our cells. And then you have these things called inclusions. So inclusions are going to be important. That's one of the things I wanted to get to. Because with inclusions, these guys allow for like storage of certain compounds or chemicals. They also allow for certain processes based upon what the inclusion is. And so the cytoskeleton is just the cell support. Skeleton. You got a skeleton. I have a skeleton too, right? And it gives me structural support. So I'm able to stand and move and all this good stuff, right? So with that, cytoskeleton is cell support. Alright, and it also can be for cell division, cell shape, growth of the cell, as well as DNA movement. So when talking about DNA movement, that's going to be for mitosis, right? And also for binary fission. So mitosis for eukaryotes, and then binary fission for our prokaryotes. Alright, so when looking at the nucleoid, so the nucleoid is where we're going to see the bacterial chromosome. So the bacterial chromosome is a circular single strand or circular thread of double-stranded DNA, right? A single strand of that double-stranded DNA that contains the cell's information. And so it's not inside a membrane, and it does not have histones. Remember, histones are involved in the packaging of the DNA. They don't have that. They don't need to package. Don't matter. Doesn't matter. All right. And so they also have plasmids. I wanted to get to this. Plasmids are important. All right. So plasmids are separate from your cell's DNA. The cell's DNA. These are extra chromosomes. So they're in addition to the chromosomes. All right. They're in addition to the chromosomes. And so they actually have genes though. They're extra chromosomal pieces of DNA. They can have 5 genes. They can have up to 100 genes. You know what we're talking about. So when looking at these plasmids, they carry non-crucial genes. So basically, they don't need the genes for survival, but they can be important. So some of the genes can encode for antibiotic resistance. That's the problem, right? So let's say, for instance, I have a bacterium that was originally not resistant to vancomycin, but then this bacterium picked up a plasmid that has a gene for vancomycin resistance. Now it's going to be resistant to vancomycin. So that's something that we actually see where plasmids can cause the cell's behavior to be changed, right? The cell's makeup to be changed, because of the way you express certain proteins, right? And so it does replicate independently of the chromosome, and as I mentioned, it can be transferred to the bacteria. It can be transferred to the bacteria. Now, this is one thing that's really, really important as well. Ribosomes, guys, ribosomes. So we know ribosomes are important for protein synthesis, and they're made up of protein and ribosomal RNA. Proteins and ribosomal RNA. Now, looking at the ribosomes, we have a 70S ribosome. Remember I told y'all that when we were comparing and contrasting prokaryotes and eukaryotes? So prokaryotes have 70S ribosomes. We can't do math here. We're not doing math. Alright, because I know when I add 50 and 30, I get 80. Do not do math. No. Alright? So here we're doing the fact that you have a 70S ribosome. Remember the S you said was the sedimentation rate, right? It's actually the Swedberg. That's the first thing. So the 70S ribosome has two parts. It has a small ribosomal subunit and it has a large ribosomal subunit. The small ribosomal subunit is the 30S component. The large ribosomal subunit is the 50S component. Alright? Now what's really cool about it is that there are some antibiotics that can interfere with protein synthesis by just targeting just the 50S subunit or the 30S subunit. Alright? Which is some pretty cool stuff. So we're looking at that. You have your gentamicin, which can target the ribosome synthesis, streptomycin, erythromycin, and chloramphenicol. Those are antibiotics, right? All antibiotics. Funny thing is, I'm allergic to erythromycin, so I'm like, dang. So since I'm allergic to erythromycin, can't take streptomycin, and I don't touch gentamicin either. Right? Let's look at the ribosomal subunit. So here we have the 30S, little small part that's going to go and combine with the 50S to make a complete ribosomal subunit. So like the proteins, well, proteins will be made when you have your mRNA. Your mRNA will come in and make all those proteins. We'll learn about those later. Alright, but I want to get some inclusions. Okay, so what are inclusions? So, when talking about these inclusions, they can hold nutrients as for when they're needed on reserve. Right? They hold them on reserve till we need it, or till the cell needs it. So metachromatic group granules, these are going to be phosphate reserves. They basically hold phosphate until the phosphate is needed by that particular cell. You have your polysaccharide granules. These will hold energy. That makes sense. So polysaccharide sugars, right? Something about some sugars. So these guys will hold energy in the form of those sugars. Lipid inclusions will hold energy in the form of those lipids. Sulfur granules will hold energy for those in the form of sulfur. And so carboxysomes. So this is going to be important for our photosynthetic organisms. So carboxysomes are important for photosynthetic organisms because Rubisco is going to be important for photosynthesis, right? The Rubisco enzyme is really important for photosynthesis for making carbon, or for allowing carbon fixation. That's one of the critical steps for photosynthesis. So this inclusion is like, oh yeah, this is great for photosynthetic organisms, right? So I was kind of disappointed with photosynthetic organisms. And then gas vascules, these guys are going to have protein cylinders that maintain buoyancy. So the ability to exist in the water environment, aquatic environment. And magnetosomes are going to have iron oxide inclusions, and they're going to basically be able to break down and destroy hydrogen peroxide. That's a good thing, because you do not want excess H2O2 in your cells. Alright? Excess H2O2 can be toxic, right? So that's a good thing. You want to have those present within those cells. Now the messed up part about it is that these bacterial cells will ultimately fight or target certain structures within the human body to compete for resources, right? And it's great for the bacterium, not so much the host. They're vying for certain resources. This is just a picture of a magnetosome, and it shows this little body of the magnetosome present in a, that's a gram negative, alright? And so then we have our endospores. Alright guys, so endospores are pretty important because it's hard to kill them. It's really hard to kill these guys. So what happens is you're a resting cell. The regular old resting cell, doing its thing, providing, getting its nutrients, living life, right? And so when you have this resting cell, it can contain a endospore that's produced inside of it when nutrients are depleted. Alright, so when nutrients aren't available, nutrients are accessible, they just can't get to them, right? And so with the endospores, once that endospore forms inside that cell, it makes the cell very resistant to a lot of things. So you can't kill that cell like talking about it because it's resistant to desiccation, which is drying out. So for desiccation needs to dry out something, to dry something out needs desiccation. For heat, chemicals, and radiation. That's why if you have an endospore, a bacteria that contains endospore, you gotta autoclave that bad boy. Alright, you gotta autoclave it. And so sometimes that's not feasible. We'll talk about that. So these guys can survive in a dormant state for thousands of years. Dormant means that it's not doing anything. It's all out latent until activated by some type of stimulus. So for instance, I tell people, for me, you had chicken pox? You had chicken? Oh, you didn't? That's good. That's good. You did? I had it. So with chicken pox, it's a type of infection that can lie latent or dormant in the body until activated by some type of stimulus. So if it's reactivated, it'll be shingles. It'll be great. And so with that, with the type of like reactivation thing, that's what I mean by that. Like it will stay, it can hang tight upon activation. Alright? And so with the endospores, this is a survival mechanism for them. It's not gonna help them make more copies, it just allows them to thrive in certain conditions. So what are the bacteria? I want you guys to know the bacteria that are gonna contain endospores. That's gonna be Bacillus and Clostridium. So Bacillus thuringiensis, anthrax, the one I told you guys, which is the bioterrorism weapon. Clostridium, you got C. difficile, Clostridium difficile. Another type of endospore infection, you have Clostridium tetani, tetanus, botulinum, right? So those are all examples of bacteria that contain endospores. But I want you guys to know the genre Bacillus and Clostridium. So to see the spores, we're gonna have a process called sporulation, where you have endospore formation and germination, when it goes back to its regular old vegetative state, resting cells, just chilling, hanging out. And we're talking about the endospores, these are important to the food industry, right? So that's why it's very important for us to kill the pathogens through pasteurization, so canned goods, right? So when talking about canned goods, a lot of times they'll tell you if you open something that's canned, do not put it back in the fridge after you open it. Do you guys do that? You open something from a can, and then you put it in the fridge, and then you're like, I'm gonna get it. To be honest. Old people do that. My grandmother used to do that. My grandmother used to she used to use evaporated milk, and you know the evaporated milk, a little small, she would actually put that in her tea. And what she would do is she'd cut like a little small hole in the side of it, and when she cut the small hole in the side, she would just, fix her little tea in the morning, add her evaporated milk to it, and just put the evaporated milk back in the fridge. I'm like, oh, she could have killed us. So the thing is that she could have killed us because what happens is if that particular can had been contaminated with Clostridium botulinum, we could have got that Clostridium perfringens. That's the one. That's the one that causes really bad diarrhea. She wouldn't have killed us. We just would have got sick. We wouldn't have died. We would have just got really sick. But really died, that's why you have pasteurization because of these guys right here. You have to pasteurize those. Working with those, milk also has to be pasteurized. That's why people when they have if they have cows and they have their own fresh milk, they have to drink it within a certain window. Right? Like raw milk. Yeah, raw milk. Some people drink their raw milk. I'm not drinking raw milk. That's just my thing, So it just depends for some people, they'll do it. I'm just not saying doing it because we need to pasteurize it. She should have did it. She should have poured the milk in a container. A whole other container. She should have been just put the milk in another container, put it in the fridge. But Alright, so with that, endospores, that's what they look like on the inside of rod-shaped bacterium. We're gonna look at them in lab. So in our lab, in a couple of weeks, we're gonna look at endospores. You're gonna see a sample of old bacterial cells, old bacterial cells, bacillus, and you'll look at new bacterial cells for bacillus. And the reason why we do that is because, remember, we said that the endospores will form when the nutrients are depleted, right? So we have a fresh culture of the bacillus. We have nutrients present. Should I see endospores? Nope, you won't see endospores in a fresh culture of bacillus. So if you have like a bacillus that's about a week old, nutrients are gonna be depleted at that point, right? They've utilized all of the nutrients in their medium and they're gonna have endospores. So we'll look at that, an old sample of bacillus versus a new sample of bacillus. Alright, so the last couple things, I'll let you guys, so looking at this, we're gonna compare eukaryotic cells, prokaryotic cells. I do want you to know what flagella are, what cilia are. I've detailed those out in the video, the glycocalyx structure, plasma membrane, all that good stuff, cytoplasm. Okay, so this is what I wanted to get to before we cut it. So So remember, prokaryotes have what type of ribosomes? 70S ribosomes. Okay, so prokaryotes have 70S ribosomes. Eukaryotes have 80S ribosomes. And their 80S ribosomes have a small 40S subunit. So that little small unit that goes with the big subunit is a 40S subunit and a 60S subunit that will be combined to make an 80S again. No snap. And so with these ribosomes in the organelles for chloroplast and mitochondria. So remember, chloroplast and mitochondria are very unique organelles. I explained it very thoroughly in the video because with the chloroplast and the mitochondria, these guys have 70S ribosomes, like prokaryotes. Alright? So that kind of tells us that mitochondria and chloroplasts came from, they have prokaryotic origins. Alright? Because they have a lot of similarities. They also have double membrane structures that kind of supports the idea that they're retaining up by the eukaryotic cells, and they pick up the membrane. So they have two membranes because they were taken up. Right? They also have their own DNA. So chloroplasts have their own DNA. You ever heard of mitochondrial DNA? Right? Mitochondrial E, where everyone has the same DNA from the original source. So mitochondrial DNA is passed from your mom. You won't get mitochondrial DNA from your dad only because of on the sperm cells have the mitochondria on the tail end. And so once the sperm penetrates the egg, the tail breaks down. And the mitochondria on the tail breaks down too. So that's why you don't get So that's why you don't get the mitochondria from your dad because it got destroyed when it was penetrating that egg cell. So, any question? Alright, so the nucleus, endoplasmic reticulum, the Golgi apparatus, mitochondria, the chloroplast, organelles, Alright, so this is what I wanted to wrap up with, the endosymbiotic theory. So the endosymbiotic theory explains the origin of eukaryotes. So this is what I was just talking about. It's the idea that larger bacterial cells were engulfed by smaller bacterial cells, right? So a large bacterial cell consumed a smaller bacterial cell. It didn't destroy it though, it just retained it, right? It retained it. Didn't break it down, it kept it. And so this nucleus formed as enfolding of the plasma membrane, and it enclosed around the chromosomes. And they believe, in theory, that the photosynthetic bacteria were ingested, and they became the chloroplasts, which makes sense. Makes a lot of sense. And then your aerobic bacteria were ingested and became mitochondria. So when you think about it, this is what they pictured for the last couple centuries. So you have the idea that you have this early cell, and not a couple centuries, I think it was the last century. So you have an early cell that was originally going to be developing into your archaea. You have your bacterial cells, and so when we look at this bacterial cell, these brown ones are going to be aerobic. They're going to have oxygen present. The green one is going to be photosynthetic. It's going to have the ability to photosynthesize. And so this photosynthetic one, or should I say the mitochondrial one, the one that was aerobic, the aerobic one was taken up by this early progenitor of your eukaryotic cell. So you have a eukaryotic cell that develops also, and as time gradually evolved, time went by, you have your nucleus forming, so your nucleic acid has a membrane around it now, you have a nucleus, and then your mitochondria is taken up, so you have mitochondria, double membrane bound organelle, you have your chloroplast taken up, double membrane bound organelle as well. Alright? So there's a couple of things that support this idea though. This whole slide right here supports the idea of the endosymbiotic theory. So the fact that mitochondria and chloroplasts, they really do look like bacteria in their size and shape. You just thought they look rod-shaped, right? They look rod-shaped like the rod-shaped bacteria, but it's rod-shaped bacteria. They also have circular DNA. Do eukaryotic cells have circular DNA? No. So eukaryotic cells have linear DNA. Prokaryotic cells have circular DNA. So this supports the idea too that mitochondria and chloroplasts came from prokaryotic cells. Both have circular DNA. Another thing is that they can reproduce independently of the whole cell. They don't even need the whole cell to divide. They just be out there dividing mitochondria here, mitochondria there, mitochondria everywhere. Same thing for the chloroplasts if they're there, right? Only plants have chloroplasts, right? So plants and some protists. We'll talk about that. Some protists have chloroplasts. And so they also have ribosomes in the same or similar size and structure to those of bacteria. So we said that the bacteria have what kind of ribosomes? 70S. 70 yes. So the mitochondria and chloroplasts have what kind of ribosomes? 70 yes. And then both have double membranes. Both the mitochondria and the chloroplast have double membranes. Alright, so I wanted to wrap it up only because I wanted to leave room for questions. Let me make sure that your brain is not up. I'll have a whole brain. I don't hurt you. Anything? Not on purpose. Now, with the study guide, guys, as I told you before, flesh that bad boy out. Right? Flesh it out. Go through it, detail it. I would say the first chapter is going to be key. The first chapter is honestly the easiest set of questions for you. Very straightforward. The only thing that I can see would be your prokaryotes and eukaryotes isn't really that hard. I broke it down. Right? So the first chapter was one of my hardest chapters in the past because I left it too big. Right? I went back and changed that for you guys. I made it to where I said, know these people. Right? So were there any questions while you were going to study that? Is there anything that popped out? You need to explain something? Because I will. Eddie, right?
Yeah. So for the chapter one, when it comes to people, example for like Johnny and Francesca, like honestly remember like the year that they
No, never years. So moving forward in our class, we know years for one thing. What's the one year we know? The golden years. What's the 18? Okay. 1857 to 1914. That's the only years I'll ever make you know. Nothing else I care about. I just want you to know what they did.
can know the names on the list, right?
Just those people. The other people, they're cool. All the other people, they're great. I just can't touch on everybody. That's not realistic.
Maybe like more details or just kind of short, like lemon, antibiotic,
So-
penicillin kind of thing, or
Yeah, so for a couple of them, I would say some more details, right? So let's hit it real quick. Paul Ehrlich, he don't even have a lot of details. It's real simple for him, right? He's my guy for the end of my guy for Salvation. Cool.
yeah.
Alexander Fleming, no details. He's my guy for what? Penicillin. All right. Edward Jenner, he's our guy for vaccination. See what I'm saying? It's really just that simple. Robert Cobb, he's the one that did a lot of stuff. My guy did a lot. So no, he did a lot of stuff. Just prove the cause. He did that through the cause posture. Yeah, he did. He was able to say a specific microbe causes a specific disease. He did. So it's only a couple people that's really detailed. Listen, Robert Cobb, got detailed. Louis Pasteur, very detailed. Everybody else though could be a one. Don't say that. I was gonna say one liner. Don't do that. Just know things.
Can you repeat again which ones were?
Really detailed. Robert Cobb and Louis Pasteur. They're both very important because Pasteur is actually considered the father of microbiology. So he's really important. Right, but the other ones could be like simple, 2 sentences maybe for the other ones. Like, did they support spontaneous generation? Did they not? Type stuff. Yeah. So, anything else? And I will tell you guys, so I won't be available this weekend. I'm being home for the most part, but my laptop's gonna be with me though. So I have my laptop with me. I have my phone with me. So if you have questions on the weekends, I got you. If you have questions, just anytime from even normally on Friday nights, I'm watching movies. But I'll leave it out for you guys. I'll leave my laptop open for you.
Just out of curiosity, are all links gonna be on it? Or
Yeah.
just kind of
No,
random?
no, all of them,
Like,
they're
maybe
really-
like 5.
So they're really gonna be on there. They're gonna be on there in some capacity. They can be a multiple. They could be a multiple choice answer. It might not be the answer, but you want to know who it is, so you
But they're all
select the
gonna
wrong
be there.
answer. Oh, they're all there.
Okay.
Oh yeah, I made sure of it. I only gave you the ones that I saw on my.
I'm just like curious since it's gonna be the random selection of.
No, I wouldn't. I don't wanna waste your time. So that's why I just gave you the ones that I know for a fact I saw. But they may not be the answer, but they're there. That
Right.
makes sense.
Mm-hmm.
Well, she was first. And I got you.
For the second one, the jumping, does that have to do with the spontaneous generation and the biogenesis?
So when talking about that one, yes. So that's what you want to include that.
I just wanted to make sure, as long as we study really well the study guide, we will pass this test.
Well, no, I can't say you're gonna pass.
Well,
I
we
think
study
you can do
no
well.
good.
You can do well. You can do well. I can't just guarantee a pass.
No, I mean, that's what we need to really look at, is the
If
study
you
guide.
honestly, okay, so I had a student. I have a lot. Last semester, they really knocked it out the park because they really studied well. They really created thorough study guides, and if you really flesh it out and you really know what you're talking about, you'll do fine. You'll do really what you learned this semester, or this exam. You have most of it is multiple choice. So most is multiple choice. You only have like 4 short response questions, and they're really not that hard because I literally like talked about it. Like, I made sure stuff that I emphasized in class. I was like, highlight this, underline this, and then the bonus. You're gonna probably laugh when you see the bonus. Because I know she didn't do that. So we talked about it. I always talk about it. And then when you think, you see it, you're like, dang, I wasn't even paying attention because she didn't say the underline that. But we talked about it. For bonus. I don't.
Trying to do the microscope and knowing that, where everything is.
Oh, you're not identifying parts of the microscope on this one, only because, so I only want you to know functions here, we only identify in lab. In lab, you only identify in lab, here's just functions. Lab is like, show me this, that, this, and this. This is just function.
So just, that Scantron or something.
So, yep, this is your Scantron that you guys will need to get. You can get it from the front office, or you can get it from, it's 50 cents. I'm saying you can get it, it's 50 cents in the front office. If you need some financial support, I know it's 50 cents, everybody don't have 50 cents. I get that. You just come talk to me, like. So, you can also get it on the main campus, that's also an option. Okay? I don't even carry cash. I only got 50 cents because I gotta change in my car, like, I don't carry cash. So,
Can you go over the amino acid structure?
yes, I can. So, with the amino acids, the structure is really cool. So remember, you got a central carbon, right? You got a central carbon, you got that little measly hydrogen just hanging out over there. You got an amino group over here, and then you're gonna have your carboxylic acid, your carboxyl group right there, and then you got your R groups. That's what's gonna be different between each amino acid. I'm not gonna make you know the amino acids, just the basic structure for the amino acid. This is called an alpha carbon, because it's a central carbon. Yeah. And sometimes you can see it written out like this too, right? Because it's still 2 hydrogens. It's still 2 hydrogens. It can be written out like that too. It's still 2 hydrogens though. Right?
And you know that. Building on the block.
Oh, yeah.
For a second, I have to figure out what the.
Yeah. Building blocks, like, build like Legos, right? Like little parts that go together and they got like a big thing. One more question, please?
So just know that they have like amino group and a carboxyl group and a cyclic.
And the hydrogen too, don't forget about
Okay.
it. It's easy, but like it's still there. No, don't count the hydrogen. Don't count all of them. Just like amino has just this little NH2, this 2 little guys. Carboxyl is just, COOH. Hydrogen hangs out over there. I mean, if you count them, that helps you, but just don't make extra work for yourself. If that's what works. Alright, any other questions?
And the R group is just gonna be.
Just R, I'm gonna literally just put it R. I'm gonna leave it R, cause I don't have time to play with you. Yeah, I'm not gonna make you do that, that's too big, I'm not doing that.
I think you answered this already, but for the exam preparation sheet, we can type it out.
You absolutely can, you can type it, write it, as long as you can read it, that's all I care about. And if you wanna, like, I was like, can I bring a handheld microscope? You gotta make it that small, like, you should work on little handheld, little, like, the little one that's little, Inspector Gadget. Yeah, magnifying glass. Magnifying glass, like, that's just, but yeah, you can type it up. So no magnifying glass. No magnifying glass.
Hey, just to be clear. Can you just like write, what was down?
Amino group?
Yeah, oh, that's the, amino carboxy and everything.
This is amino group. This is the carboxyl group. This is the R group. Hydrogen, carboxyl, amino. Alright guys, so we're on to lesson 5.
Yeah.